So thank you so much, Eben, for kicking us off. We wanted to have a book series celebrating the books of the different authors that we have such a large volume of books that are being produced in ADI, and we rarely get a chance to hear about all of them. And your book especially was one that we were really curious about and really wanted to know more about and celebrate because of just what a compelling issue it addresses, how brilliantly it has done, having an impact already in its very early stages. So very briefly, to introduce Associate Professor Eben Kirksey, an anthropologist and an associate professor of research at the Alfred Deakin Institute. And Eben's been really very widely published in very high profile places. Just to give you a very small sample of these, it's The Atlantic, The Guardian, The Sunday Times. Duke University Press has published 2 of his books, Freedom in Entangled Worlds in 2012 and Emergent Ecologies in 2015, as well as 1 edited collection, soon to become 2 edited collections, I think. And Eben is best known for his work in multi-species ethnography, a field that has had such a tremendous impact on so many of our works, including my own. It's a field that situates contemporary scholarship on animals, microbes, plants, and fungi within deeply rooted traditions of environmental anthropology, continental philosophy, and the sociology of science. The Mutant Project, Inside the Global Race to Genetically Modify Humans, is Eben's latest book, and it visits the frontiers of genetics, medicine, and technology. And it fundamentally asks whose values are guiding gene editing experiments, and what does this era of scientific inquiry mean for the future of human species? These are really tremendously large questions, and I hand over to Eben to address these in his talk. Thanks,
Yamini. Thanks so much for having me. And I'm just going to sort of maybe informally tell some stories today from the book, try to draw out some of the key ideas and introduce you to the places and people that I encountered. The story begins in Hong Kong, where I was checking into a hotel right ahead of this summit. So we were delegates from all around the world that were coming together to talk about the promise and peril of using genetic engineering technologies on the human species. So, many tools have been tested out for decades now on animals and microbes and plants. And a consensus was growing that there's new medical applications that need to be addressed, including applications in future children. So as I was checking into this hotel, news was breaking, and theory had just been put into practice. So someone staying in the hotel, Dr. Jiankui, had just announced on YouTube moments ago that 2 genetically modified children had been born. He was awkwardly sitting on the couch here in the lobby, trying to explain himself to Jennifer Doudna, who later went on to win the Nobel Prize for her pioneering research on CRISPR. CRISPR is a new, fast and cheap genetic engineering tool that can, target DNA in a specific kind of way. And he was trying to explain himself, and meanwhile, this is going viral. The YouTube video is getting shared all over the place. At this point, some people are speculating it's a hoax. Reporters start swarming. Usually academic conferences, you might have a smattering of participants in the audience, but this was a swarm of reporters, and so we all had our PowerPoints and basically had to think on our feet and address this new fact that had emerged, even as we were trying to figure out what was going on. So in some ways, this moment was a recapitulation of an earlier moment in scientific history. So this is a photo taken of Louise Brown moments after her birth. And Louise Brown was similarly controversial. Her birth was condemned by everyone from the Pope to members of the scientific establishment. The medical researchers pictured here, Steptoe and Edwards, had their laboratory shut down for a number of years and were only able to later start it up with private funding. Now IVF is ubiquitous. It's very much accepted clinical practice for helping couples with fertility problems have children. But in those days, it was seen either as a miracle or as some kind of radical aberration that totally disrupted what it means to be human. But that settled, and what we have now, many years later, is a technology that does predictably help parents, but it's actually not that efficient, and there's problems with it. We can talk about those existing problems with IVF later if people are interested in it. But the point is that it wasn't miraculous. This birth was just kind of an incremental advance in science. Dr. Ha was very much trying to follow in the footsteps, and he very much wanted a photo op, very similar to the picture of Louise Brown. So this is part of the participant consent form that patients in Dr. Ha's CRISPR trial signed. So here the parents were giving consent for a picture to be taken moments after birth, and explicit consent relating to where those pictures might appear, everything from elevators to outdoor billboards, books, calendars. Clearly there was an entrepreneurial spirit guiding this. This is the announcement.
Two beautiful little Chinese girls named Lulu and Nana came crying into the world as healthy as any other babies a few weeks ago.
So that video and a few others that he posted on YouTube was basically all we had to go with as the summit started, and as we tried to make sense of scientific history in the making. Later, after the summit, Dr. Ha was detained by Chinese authorities. He was escorted back to mainland China by the president of his university and members of the security forces, placed under house arrest for a while. Then he was rendered to a black site and eventually charged with 3 years in prison for medical malpractice. I was able to later gain access to key individuals in the lab, as well as documents, and tell part of the story that was totally untold. And one of the significant things that I reveal in my book is that these children were actually not healthy at birth. They were born at 31 weeks. They had trouble breathing initially. And at the time that YouTube video was released, one of them was still in the neonatal intensive care unit. So the book goes into a big open question. Was this premature birth? Were the health problems experienced by these children at birth a result of known issues with IVF? Many couples who've gone through IVF know how hard it is to use this technology. Anyone who's had twins know that twins are much more likely to have a premature birth and other issues at birth. But this was part of what was not being discussed at that summit, and that has only now come out with my book. Dr. Ha had support of his university. He had support from a lot of biotech investors. He also had support from key members of the Communist Party, officials in Beijing, as well as Shenzhen, the city where he was operating. Shenzhen is very much like the Silicon Valley of China, driven by these values of disruptive innovation. The mantra there is move fast and break things. And this is partially what CRISPR does. We can talk a little bit more about CRISPR later, but basically it's a tool that breaks DNA. It grabs a hold of the DNA double helix, it produces a break, and then the cell tries to repair the damage and usually botches the job. So that's when you talk about deleting a gene, what you're actually doing is producing a targeted mutation. But some of these quotes here from social media illustrate that the Chinese public really wasn't approving of this experiment. He anticipated some degree of scientific controversy about what he had done. He anticipated having to ask and answer some serious bioethical questions, but he wasn't prepared for the tidal wave of public opinion condemning this experiment. A few years earlier, another researcher from southern China, Junjun Huang, had used CRISPR to modify human embryos. And the key difference between Dr. Ha's experiment and Junjun Huang's experiment is that those earlier embryos weren't implanted. And at that point, there was a huge controversy in the international media, but the Chinese media didn't really react, and neither did the public. And I think the differences in reaction between these two consequential experiments relate to fundamental differences in ethics and values. In China, there was a sense of kind of secular progress being the guiding norms and values of society ever since the Cultural Revolution. Chairman Mao very much tried to get religion and other sort of superstitious beliefs, is how he termed his kind of modernist campaign. He wanted to get rid of that and establish a modern project for China that was founded on different values related to technology and progress and social engineering. But really, I think you see the persistence of deeply held ethical values in China that are from a variety of religious traditions. So you do have the principles and values of socialism and Marxism, but you also have Taoist principles. You have ideas from Confucianism that regard a person as a person at the moment of birth, not at the moment of conception. So, it's an ethical landscape where it's not necessarily Christian bioethics that's governing how people respond to and react to experiments like this, but it's a different sense of personhood. So the stakes of these ethical conversations are different. Some scientists on the sidelines of the conference in Hong Kong that I attended said pretty much racist things, like there are no ethics and values in China right now. And I came to understand this experiment in context. In a academic and regulatory setting where the conversation is less about bioethics in a Christian moral idiom, but it's about a medical ethics where prudence, doing the right thing at the right time, caring for patients, improving the well-being of people, those are some of the goals and mandates. So different ethical systems. But again, I think it's the public reaction that really derailed the trajectory that Dr. He imagined himself on. He, in a moment, went from being one of the most famous scientists alive to experiencing a dramatic fall from grace. And that dramatic fall, again, wasn't just scientific controversy, but these social media discussions. So, 250 million views of this hashtag, 1.6 billion interactions with this other hashtag. This was a moment in the consciousness of China unlike any before. So I think when in sorting through all the diversity of opinion that was registered on social media. So WeChat is something like it's a messaging app, but you also use it to pay for things. It's sort of like Facebook maybe, or Twitter. Weibo is more of a bloggy kind of Facebook platform. But if you sort through all the comments that accumulated on these hashtags, I think a lot of them were just concerned about the babies and concerned not only about what kinds of impacts the science might have, but others started posting things about how they're going to be surveilled for the rest of their life by Chinese authorities. Again, the health problems at this moment were totally unknown. This is only something that became public last year with the publication of my book. And what came out as this controversy settled down, as the case went to trial, is that the parents actually supported Dr. He, in some cases enthusiastically. So I was able to get court filings that the parents made, letters in support of him. And the parents simply desired to have a healthy baby. And this is something we can come back to in the context of this experiment targeting HIV and the extreme stigma experienced by the parents and by the men in particular who are HIV positive. So understanding Dr. He's story involves going back to his humble origins. So he was born here. That's his house pictured in the background over those trees. And this is a rural part of the Hunan province. parents. His parents made around $100 a year. That was their annual income. And I heard the story of his birth. I talked to the woman who wasn't a midwife, a family friend, who ushered him into the world. And he came out with the umbilical cord wrapped around his neck. The hospital was about an hour away, first by bus, and then, first by walking, and then by public bus. And there were no telephones, no electricity in the village at this time, no roads. So this is, 1984, relatively recently, but a moment in time where there's profound inequity, where China very much feels like a developing country. So as he grew up, the experience of electrification of the village inspired his brother to become an engineer. He became an electrical engineer at the hydroelectric plant. And the family basically helped him in a modest way, sort of work his way through school. He became a star pupil at the Xinhua High School. Xinhua is a magnet program in this province. And from there, he went on to a prestigious national university and eventually to a PhD at Rice University and a postdoc at Stanford. So he was recruited from Stanford, the heart of Silicon Valley, back to China, to Shenzhen, a part of the country that is known for a characteristic speed, doing things faster, not only faster than anywhere else in China, but faster than anywhere else in the world. The idea of Shenzhen speed emerged in the 1980s, where buildings were going up, skyscrapers at a pace that was, eclipsing everywhere else. And the idea of Shenzhen speed has been brought to electronics. So, the phone that you're likely carrying around your pocket, the computer that we're interfacing with right now was likely manufactured in Shenzhen. It used to just be cheap plastic toys like Cabbage Patch Kids and boom boxes that came from Shenzhen, but now it's some of the more innovative technological products. And you're seeing an increase of patents emerge from this part of China, very much outpacing the speed of technological innovation in the US and other parts of the world. So it was part of this entrepreneurial culture, part of this culture of innovation that is the background for this experiment. And the university, this is the hospital that gave him ethics approval for his work. And initially, when I went there, and I tell the story in the book, I thought this is where the clinical work was done as well. But it turned out to be a very complicated project involving multiple institutions. And I reveal at least as much as I'm able to in the book about all the different institutions involved. His university was very much involved. This is where Dr. He met with the patients who consented to participate in this experiment. This is where the genomics work was done, where he had bioinformatics technicians studying the genomes of the couples that created embryos for the experiment. And it wasn't just the one couple. So if you follow this case, you probably heard about a third baby that was born after the twins, who were named Lulu and Nana by Dr. He. But there was actually, I believe it was 7 couples, I'd have to go back and look at the book, who created genetically modified embryos and had those implanted. But most of those embryos didn't take. Some of the other facilities involved were not in China. The book describes how Dr. He traveled together with a colleague from New York City to Bangkok, where some of the first implantations of CRISPR-modified embryos were conducted. I also describe how that same pair, Dr. He and Dr. Zhang Zheng, went to the Hainan province of China. It's sort of like China's Hawaii, but it's also a new medical tourism zone. So they met with some of the governor's key staff members and were actively discussing a proposal to open up a new clinic with 100 doctors. They toured a facility. And even at the moment when the health of the babies was unknown, when his technicians back at the university were still trying to figure out basic questions related to, did our experiment create unintended genetic damage in these babies? Are their health problems a result of our experiment, or is it a result of IVF? even at that moment, Dr. He was traveling to Hainan, to Thailand, to Beijing, to California, trying to get new investors on board. So in thinking about some of the fundamental values driving this experiment, it was very much the values of the entrepreneurial economy, values of speed, values of innovation, values of disruption. And what you saw as the trial took place, as the Communist Party reckoned with this transgression, was that some of the older values of China came into conflict with these newer values, where old members of the party, the old guard of the Communist Party, were insisting, what we need is more prudent science. What we need is better regulation. What we need are, there's lots of discourse about the gentleman in Chinese society who does the right thing at the right time. This clearly wasn't the right thing at the right time, and he was put in jail. But at the same time he was put in jail, China has announced new funding opportunities and new regulations funding this kind of research going forward. So it's not that the use of CRISPR in human embryos is going to be stopped in China, but it's going at a slower pace with more, sort of thoughtful deliberation. And we can talk about, whether this should be happening at all. So part of the backstory that I explore in the book is the stories of the parents who signed up. So it's also a story of HIV. So in China, like in Australia or the US, HIV isn't so much of a medical problem anymore, as long as you take your medicine. And China has an innovative social program for HIV patients that guarantees them free medication for life, free counseling, free testing, although viral load testing, I've found out, isn't as easy to get as one might like, especially if you're trying to have a child. But at the same time, at least compared to a place like the US, where the out-of-pocket expenses for an individual or for an insurance company that's helping an HIV-positive person stay on medication, those expenses in the US are about $30,000 a year. So, there is a need, in a sense, with a genetic intervention, there's a hope that there might be a one and done cure. The idea of this experiment was that we knock out a single receptor, and I'll show you some pictures of that later, and make these children unable to get infected with HIV. So there's a natural mutation that occurs in the human population, and the idea was to replicate it in these children. So for the parents, they were less concerned with the science. In reviewing the participant consent forms, I realized, and some members of the lab told me, that they didn't really understand how profoundly significant this moment might be for humanity, as humanity reckons with the world's first genetically modified children. But these parents were basically interested in having a healthy child. They were prevented from having a child by Chinese policy. So if you're HIV positive, you're thinking about infecting your partner. You're thinking about not wanting to transmit the virus to your offspring. And one way to avoid that is by using IVF, in vitro fertilization, or another technique called sperm washing. So you don't actually have to, take the embryo and egg out of someone's body and put it back in, but there's other laboratory techniques that you can use to get rid of the HIV viral particles in semen. By law, HIV positive individuals in China cannot avail themselves of those techniques. It is possible to do reproductive tourism. There's quite a vibrant industry that helps HIV positive people, and in particular, gay men who might also be thinking about surrogacy. Surrogacy is also illegal in China. But there's networks that have been established over social media and other ways to bring people to Thailand, to bring people to the United States, to bring people to places that have the medical technology and also more permissive legal environments. But these particular couples, many of them were party officials. Some of them were active duty military. So they couldn't travel outside of the country without getting special passport permissions. Others simply couldn't afford it. So the idea of having a baby without IVF, without sperm washing, was not something that these families wanted to do. So in some ways, these parents were trying to act ethically by signing up for this experiment. And there was moments that I chronicle in the book where communications broke down between the lab and the hospital that did the implantations and the patients. At certain moments, and this is the story of the third baby, parents were insisting, I want to have this implantation happen, even before they knew whether or not these first two babies were safe and healthy. So there was a lot of conflicting values, interests, hopes, dreams, and desires coming to play at this moment. But one of the overriding ones that came from the parents was a dream of an HIV future, a dream of a child that would be liberated from the social stigma of living with HIV. So, in part, this details of the experiment were kept secret because Dr. Ha was trying to, announce this after, the data had settled. He actually didn't want to release the YouTube videos at the Hong Kong conference. He'd submitted a paper to the journal Nature that was under consideration. So he was wanting to publish that paper first and then explain it to the world with the YouTube videos, but a journalist leaked the story. So it all kind of erupted at this moment. But again, for the parents, they're hoping for healthy children. They're hoping for children that are free of HIV, but they're also wanting to protect themselves. And the profound secrecy that they, experienced in, they were not only trying to sort of follow Dr. Ha's mandates to keep what he called trade secrets from the public, but they were also wanting to make sure that their bosses, that their family didn't know about their HIV status, because when the HIV status is disclosed in China, that often results in someone losing their job or in intense family conflict. So I want to get into more of the molecular details now. I've kind of given you a backdrop of some of the social, cultural, economic, political, ethical issues in play, but I want to kind of get at some of the molecular basics so you can understand what CRISPR is and what happened with this particular experiment. So this is an artist rendering of the molecule and how it works on a molecular level. So as I explained before, it basically grabs a hold of the DNA double helix and it breaks it. And it then relies on the cell to stitch it back together. And, this is where the title of the book comes from. It creates mutants. It creates targeted mutagenesis. So you can give CRISPR coordinates within the genome, and the molecule basically goes there and disrupts the genetic code. There's a lot of metaphors that people have used to talk about CRISPR. The dominant one is editing. With editing software like Word, you can type a sentence. If you make a spelling mistake, you go back and correct it. You can cut, you can paste, you can do all kinds of things. But that's not actually how CRISPR works. As I studied the molecular details, I learned that it works more like a drone that has missiles. So you give a drone coordinates, sometimes it takes out the target. This is the drone warfare of George Bush and Obama and Trump. You can give a drone with Hellfire missiles coordinates, and sometimes it takes out the so-called terrorist, but also sometimes it takes out the wedding party. And sometimes the drone goes astray and gets the coordinates wrong and hits some other target. And this is how CRISPR can work. And maybe it's, a drone isn't a perfect metaphor either. This molecule can bounce around in the genome up to several weeks after the intended target is hit. So it can create off-target damage. And this was in part what Dr. Ha's lab was looking for after this experiment took place. The particular receptor that they targeted is called CCR5. So on this screen, you can see the HIV virus pictured here in pink, and you can see a couple of different receptors. So receptors are basically little sort of locks, I guess, is one metaphor. And if the virus, you might have heard about, spike and ACE2, those are the receptors with COVID. But this is, HIV has some surface proteins that bind to CCR5, and then that basically transports the virus into the cell, and that's how it infects it. So the idea was that you would knock out CCR5, and that these cells would be totally resistant to HIV. The story with HIV is complicated. It turns out there's co-receptors involved. CCR5 isn't the only receptor. There's another one there that you see called CD4. So there's some strains of HIV that can actually get into cells even in individuals that naturally have the CCR5 receptor missing. But Dr. He thought that it would be a relatively simple technical matter. He was a biophysicist. He wasn't a geneticist. He wasn't a doctor, a physician. This is why he was charged with medical malpractice. He was conducting medical experiments without a license, but he thought it would be a simple technical matter. You just take out this receptor and the virus won't get in. So my book explores the experience of other patients who also had this receptor deleted from their genome, but it was a different kind of experiment. So this is Jay Johnson, and he's one of the world's first gene-edited people. He participated in the very first gene editing trial 10 years before this experiment took place in China. The experiment that Jay participated in wasn't with CRISPR, it was with another tool called zinc fingers. And Jay is part of an HIV positive community that has lived with the virus for decades now. Jay watched his friends, some of his boyfriends, die of AIDS during the original epidemic, and was a nurse, became a nurse and a caregiver who volunteered, after working long shifts at the hospital for paid work, he spent after hours caring for loved ones as they were dying. And Jay currently is involved in an organization called Action Wellness, an organization that is all about identifying pathways for people who have chronic infections like HIV or other chronic illnesses to get medicine. So as many of you may know, in the US, there is no national healthcare system. It's a patchwork of insurance schemes. And many of these schemes exclude people. And so Jay's organization works with individuals to make sure that they're getting HIV medicines, irrespective of their gender identity, their race, their immigration status, or their employment status. So there's basically an ongoing field of activism taking place. Jay's personal experience in getting this therapy, which was different than what happened with Dr. He, was a traumatic one. So Jay basically went into the hospital, and it's an environment that he's very familiar with. So for the initial procedure, it was relatively simple. They took out his blood and spun it through a machine called an apheresis machine that separated the white blood cells from the red blood cells. The machine shunted the red blood cells back into his body, and then the white blood cells were taken away to a lab. And we're talking about billions of cells at once. The adult body has about 34 trillion cells in it. So they were just taking a fraction of the cells from his body, genetically modifying him, targeting that same gene, CCR5, so knocking out CCR5, but doing an idiosyncratic pattern of damage each time. So it wasn't like a clean deletion, it was a series of targeted mutations. And it worked in a proportion of those cells, about 1/4 of the cells. In tests that showed that this worked. But the traumatic moment came when Jay went back to the hospital to get the blood transfusion of the genetically modified cells back into his body. And this was a completely unknown moment. No one had done this before. And as the cells started going in, Jay said that he started feeling chills. He then started shaking and then going into full rigors. And then they brought out the crash cart. And being a nurse, he knows that seeing the crash cart is really bad news. That means that you might have to revive someone who's, either had cardiac arrest or stopped breathing. So it seemed like a really acute situation. There was a cluster of researchers, doctors, nurses in the room. They gave him some Benadryl. He fell asleep and basically woke up a few hours later and was fine. And this, they later understood, was a transfusion reaction. It had nothing to do with having genetically modified cells into his body, but had everything to do with getting a sudden transfusion of a lot of plasma back in. So Jay is now participating in community advisory boards, advising researchers, drug developers, and also members of the HIV positive community about participating in these experiments. He is shaping and actively changing the ways that consent works in these kinds of processes. He's telling people in his community his story. He's emphasizing, that this was an intensely traumatic moment to get this, but he would do it again. He sees a lot of promise in gene therapy, even though he didn't get any direct medical benefit himself. So one of the other participants, and so Jay was in the phase 1 clinical trial, where they just had 12 participants. Matt Sharp was in phase 2. And Matt has a slightly different history than Jay. Jay his response to the HIV epidemic was to engage in care work, to engage in advocacy work, to engage in organizing work at kind of an NGO level. Matt Sharp was an activist who was in the streets. So he participated in a number of the iconic demonstrations by this activist collective, ACT UP. And he can't remember how many times he's been arrested. So, when you talk to him, he's very quick to say, I think I've been arrested around 11 times, and I think I've been in around 8 clinical trials, or maybe it's the other way around. But from the early days, the stakes of this activist movement were clear. People were dying. The established treatment was a drug called AZT, which had very toxic side effects and very little medical benefit. So they were campaigning since the late 80s, early 90s for risky, innovative treatments to deal with HIV. So when Matt learned about this gene editing clinical trial from one of his doctor friends over a lunch in San Francisco, he did everything he could to sign up. He was really excited. I asked him about his experience during the transfusion, if he was afraid of becoming genetically modified, if he was worried about the possibility that one of these cells would have mutations that would make it malignant, that there would be some unintended consequences. And he said no. He saw the technology as full of hope and full of promise. And as I pushed, he said, let me tell you about the thymus transplant that they tried to give me in 1993. They basically took a thymus out of a baby, a small little organ where the T cells mature, cut a hole into his chest, put it back in, didn't, seem to get rejected, didn't really seem to give him any benefit. So they took it out a few weeks later. So it was a lot of blood, a lot of very invasive procedures, but little medical benefit. And so Matt's experience in this trial was very different from Jay Johnson's. Matt is a part of a group of patients who have HIV who are called immunological non-responders. So most people with HIV today, if you're taking your medicine, your immune system's fine. You have a life expectancy that's very similar to normal. And in fact, you might even have a life expectancy longer than most of the rest of the population, simply because you're going to the doctor more, you're being more attentive to your health. But there are, a number of HIV patients, up to 20% in some parts of the world, where the immune systems don't recover. So what Matt experienced after this transfusion was a profound improvement in his cell counts. So he had been hovering near AIDS. So AIDS is not HIV. HIV is the virus. AIDS is the medical condition. And that's when your immune system basically stops working, when you don't have enough white blood cells to mount an immune response to a new pathogen. So Matt's cell counts had been basically at the level of AIDS, on the border of that, ever since his first diagnosis in 1989. And with this new gene therapy, his cell counts were normal again for the first time in like 20, 30 years. And all of a sudden, this technology that had been just in the realm of hope and hype, gene therapies have been around, at least in the imagination, for decades. But, when Bill Clinton announced the draft human genome in the year 2000, it seemed like the promise of all of these new medicines would be unlocked. But at this point, when Jay's doing this trial in 2008, there still wasn't a single gene therapy on the market. So this one looked like it might be it. But basically the company, after making $1 billion on market capitalization, their stock went way up after these preliminary findings. One of the key scientists went to jail for insider trading, and then the stock crashed. And the company decided that they wanted to keep going with their gene editing work. The company's called Sangamo. but that AIDS and HIV was not a priority. So they decided to focus their resources elsewhere. So Matt Sharp and a number of other activists have now become engaged in this long-term battle with a company that modified their DNA to get new experiments started. So basically, through this lived experiment of participation in a clinical trial, he saw the hope and the promise in very concrete terms of having an immune system that was repaired, and basically wanted to get a second dose if his cell counts ever dropped again, as they have since, and also expand this to other members of his community. But the company basically decided that they wouldn't make any money off of this treatment. Since then, there have been new gene therapies. The very first gene therapies that I describe in the book have been approved by the FDA, and they're coming with price tags. The first one was $400,000 for leukemia from Novartis. Another one was $2.1 million for spinal muscular atrophy. And there's about 800 gene therapies in the pipeline right now. So these treatments are coming. So for me, as I went along this journey, the ethics kind of shifted. And I came to see that some of the core ethical questions relate to who gets access to these treatments. You know, now that we have this new generation of precision medicine, of gene therapies tailored to individual people, who's going to enjoy those benefits? And you know what we have with ACT UP and the work of Matt Sharp and many others is an ongoing social struggle, not just about developing these new innovative treatments to a disease that's very difficult to get rid of, but an ongoing struggle about equity of access, opening up questions about who's getting AIDS, how and why. How can we think about these issues alongside issues of homelessness, issues of housing precarity, issues of employment precarity? So the book covers all these options, all these different issues and many more. I haven't talked today about my work in Indonesia, London, other places where I was following, you know, the reproductive uses of gene editing and genomic medicine. And in conclusion, I'd just like to maybe pan out with a little history of ideas to situate the title of the book a little bit more. It's called The Mutant Project, and it is in part in dialogue with science fiction. So we can talk about the X-Men and the ways that a parable by Jewish American authors about eugenics, and in many ways you can, you see, like in the original X-Men movie, a young boy being taken away from his parents at Auschwitz, who later turns into the supervillain Magneto. I think this is a really interesting parable to think about ideas about eugenics, which are very much in the fore of this book. But if you think about mutation theory, it starts in 1901. So a Dutch botanist published a book with that title, Mutation Theory, at the turn of the 20th century. And at that moment, it seemed like new species could form even faster than Darwin dreamed. It seemed like mutations would be revolutionary rather than evolutionary, rather than this gradual change. The idea with that book in 1901 is that you would see a mutation and there'd be a sudden transformation. Later on, in the early 20th century, we saw all kinds of attempts by scientists to induce mutations. So there was this idea that scientific enterprises could control evolution and harness its power for human ends. And you saw a lot of experiments with fruit flies and other things and all sorts of mixed results. By the mid-20th century, with the dawning of the atomic age and fears of radioactive fallout, other concepts emerged around mutation, like genetic load. The idea that, initially mutations were seen as a good thing, as something uniformly good. But with genetic damage that happens from radioactive exposures, toxic exposures, everyone began to appreciate that genetic injuries can cause serious ongoing harm. And as a result, there were many environmental policies put into place in the 60s that tried to control these exposures. At the same time, a little bit later, Stephen Jay Gould celebrated mutation as a source of biological diversity. He argued that evolutionary change can happen abruptly, with major mutation events producing what he called hopeful monsters. And, anthropologists have studied mutation for a couple of decades now. Joe Masco insists that it's a mistake to simply celebrate mutation as whimsical possibility. He's studied mutant plants and animals that have been cropping up near nuclear test sites. So genetic mutations, as we can see now, looking back from the 21st century, they can result in new adaptations to a changing environment. They can be fodder material for random genetic drift, or they can produce critical injury. So today, as our bodies are experiencing cancer on an unprecedented rate, where 1 out of every 2 people in the United States is likely to come down with cancer, we need to be thinking about those malignant mutations. But with the logic of Isabel Stinger's Pharmacon, we can think about maybe the poison being the cure, and the ambivalence of that. So do you want to do targeted mutagenesis for some medical benefit, or, as many people are dreaming about these days, enhancement? That's another topic that I cover in the book. What are the consequences as now we finally do have these tools that earlier scientists dreamed of, these tools that let us harness evolution, that let us harness mutation and direct it. So with that, I'll open it up for questions, and thanks again, Yamini, for hosting this talk and fostering this dialogue.
Wow, thank you for that, Eben. That was an absolute tour de force, and I think you gave us such an interesting and such a exhaustive and a neatly encapsulated overview of not just the scientific and medical complexities, but just the kind of hefty political and social questions it raises, but also the business opportunities and the commercial opportunities associated with gene editing going forward. That was just absolutely amazing. So, sorry, opening it forward to questions. Would anyone like to go first? To raise a couple of questions?
I think, Stephen, is that your hand up?
Stephen, your microphone is not on.
Sorry, I was trying to find the raise hand function. That was really fascinating, just mind-blowing stuff. I guess I'm coming from an international relations background. One of the things that interests me is the whole unregulated nature of this, and how different areas are different countries are sort of playing off each other in terms of this race that you're talking about. I guess the question is there any efforts afoot to try to regulate this at a global level, or to try to kind of create some rules or guidelines for this kind of stuff?
Yes, there are. And so Australia is one of the leaders in terms of having very specific and proactive legislation about these kinds of things. India is another place where you basically can't do this kind of research. But at the same time, as this experiment was happening in China, as it was happening in Bangkok, Thailand, I also, as an ethnographer, I'm constantly listening to other jurisdictions that might be doing things. At a certain moment, a clinic popped up in Cyprus. They were publicly advertising CRISPR babies. One of the key collaborators of Dr. He Zhang operates a clinic out of New York City, but has traveled to Mexico, where he did a controversial experiment related to so-called 3-parent babies with mitochondrial transfer. And he also operates a clinic out of the Ukraine. So there is an effort underway, and I believe it's 170 or 80 odd countries have signed on to a temporary moratorium on using CRISPR in germline editing situations. So germline basically means you're doing the experiment that Dr. He did at the embryonic level, rather than the experiment that Matt Sharp and Jay Johnson participated in at the adult level. So there's a consensus among the scientific community, and very much being led by some of these profit-driven enterprises, that there's a profound promise of genomic medicine, and we need to be using this in adults. I think, that's a whole separate issue, but there's problems in just kind of forging ahead in adults and only thinking about profound ethical problems as they pertain to children. One of those is also long-term risk. I didn't mention that 2 of the 4 people I interviewed from that trial that targeted CCR5 10 years before 2 out of 4 have experienced prostate cancer. So, you know, it hasn't been adequately followed, in my opinion. And some of the participants in that experiment were only monitored for 5 years. So as we're entering these new eras and new experimental moments for the human species, I think the onus really is on the scientists and on the regulators to come up with better policies and procedures to make sure some of these ethical missteps aren't repeated. One of the things that Dr. Ha actually got right is that he got an insurance package for each of the children that would result from his experiment. That hasn't been done with these patients in the US. And there isn't really an independent body that is engaged in the long-term monitoring. So what we have right now is a very patchy landscape in the US. Similar experiments to Dr. Ha's are prevented by what's called a rider on the appropriations budget. So every year Congress asks the administration for money, or it's a conversation actually. Congress allocates the money, the administration asks for it, there's a back and forth. So part of the appropriations bill says no federal funds for this kind of research. And there's a rule in place with the FDA prohibiting them from reviewing clinical protocol related to this. But you could imagine a lawyer, as someone did on the sidelines of the summit in Hong Kong, making a case that this isn't an experiment. This is innovative clinical medicine. And that's how some of these procedures have been rolled out with IVF in the past. So I wouldn't be surprised if other CRISPR babies have been born since the 3 that I know about. The technology itself is relatively easy to use. I took a course that I detail at the NIH. I know how to design my own CRISPR targets now. All I would need is, a willing IVF lab. And, there are people who have, it took a week of my time. I mean, I had an undergraduate degree in biology, so I had the background. But the most expensive machine in terms of the CRISPR part was an Alonza electroporator, which I think is about $5,000 new, or you can get it on eBay for a couple $100 up to $1,000. So the expensive part of the machinery that you need is the IVF rig, and that is pricey. But there's IVF clinics all over the world that have those IVF rigs. So it's basically just bringing 2 fields together. So it does not take a profound degree of expertise to replicate this experiment.
Thank you. That was really interesting.
That's a bit terrifying. to understand how easy it is to use.
Maybe you can
does anyone else have a question? Sorry?
I was going to say maybe even can tweak my ability to write A-star journals. That would be good.
In part, this book is also using CRISPR to diagnose problems in genetic thinking, right? So, since the 1950s, Watson and Crick have said, we've unlocked the promise of life. Genes explain so much about what it means to be human or what it means to be other things. But, in going through this book and looking at the actual targets that people are hitting with these tools and what can be changed and what's beyond the scope of the tools, so many things are going to probably be forever beyond the capacity of these kinds of enhancements. So, for example, height. Height is something that's generally seen as something you inherit from your parents, but it turns out that there's 180 genes at least involved, and that only explains a fraction of your height. So if you want tall kids, better give them growth hormones rather than CRISPR experiment. That's true. Much less something so difficult to pin down as intelligence. There's a guy I profile in the book, Steve Su, who is working with a company called BGI in Shenzhen, China, and they've been trying to study the genomics of intelligence, and they've identified a bunch of different loci, genetic loci, that more or less correlate with some aptitude. But we have multiple intelligences, and any one of these genes, there is no sprinting gene, there's no soccer gene, there's no gay gene, it turns out. So all of these things that we were told about nature and nurture in the 1990s, like a lot of that genetic determinism is coming unraveled even as, there are some medical uses and some, so, if you want to enhance yourself, if you want to have huge muscles, like I can do it. Like we could make a baby that has massive muscles, but that baby would have heart problems.
Yeah.
So you can delete a gene called myostatin that
Wow.
produces, there's a kid on YouTube that, if you search Juliana Stroh, you can see a kid who's like 5 years old and looks like Arnold Schwarzenegger. And there's cattle that also have this mutation. It's been induced in rats. But yeah, when you take out myostatin, you have smaller organs generally and a propensity for heart problems. You could take out the gene called EPO, which would enhance your endurance, but it would make you more likely to have strokes. You could take out the genes that make you feel pain, which is a relatively simple thing to do. But kids are born all around the world with this, and most of them have very seriously reduced lifespans. Many of them bite their tongues off as babies. So, there are certain genes that you can target with mutations, but most of the complicated things like intelligence or like athletic ability, there's a project at Stanford that's sequencing the genomes of elite athletes and finding that basically each one has an idiosyncratic genetic makeup that is doing this elite, sprinting or endurance running in a different kind of way. So ideas of enhancement, I think, are going to start to fall apart as the public kind of catches up to where the science is.
Yeah. Thanks for that. Oh, Torvald, he has a question. Thanks,
yes, can you hear me?
Yes.
Oh, well, thank you very much for a very interesting presentation of your book. What interests me is exactly the balance between ethics and science, and you covered that in a very good way. I'm worried because what I've seen in my long life is that the economic influence, the big business, even the Chinese as well, have a very big influence now on what is going on in the world. Klaus Schwab from World Economic Forum has expressed a very direct interest in this area. Where do you see the balance that can keep back the economic experiments, the scientific experiments dominated by economic interests, with this very important ethic here? Seems to be very easy to find individuals that are willing to go in here, but there must be from the society of such a counterbalance to the economic interests. How do you see this balance being established?
So right now in the US and China, that doesn't exist. So if you just take the example of this particular experiment, the investors were never penalized. The international collaborators, so the PhD mentor of Dr. He from Stanford, his name is Michael Deem, he was in the room, he was on campus in Shenzhen, China, when the participant consent process took place, when these parents were asked if they wanted to participate in this trial. Michael Deem's name was on the paper that was submitted to Nature, and he was also an investor in this company. And I think we have a very explosive mix of scientific hubris and the power of what Kaushik Sundararajan and others have called biocapital, where it's all about speculation, hope, promise, and it's speculation in the sense of market speculation that is generating these bubbles of money that are astronomical. I mean, the companies that Dr. Ha founded they were all based on a promise. They weren't, he had a genomics company that, at least last time I checked, hadn't produced an actual gene sequencing machine, but it produced the promise of a future gene sequencing machine that would be better or different than the current ones on the market. And you see a lot of these spaces of promissory capital that just have, seemingly unended velocity and resources. Like this is not a story of China's Frankenstein, as some of the journalists hastily reported. this is a story of a guy who had, a corporate office of his own, Direct Genomics, with 2 levels, with 60 employees working in cubicles, working on the lab bench. And, that company still exists. Dr. Ha's divested of it. And he's become sort of a scapegoat for an industry and, scientific enterprise that continues with the same, maybe not the same velocity. Things have slowed down in this particular field, and there is a lot of introspection, but, people moved on. There's other political things that are more pressing, and engaging in the work of anticipatory governance, anticipating where these scientific projects are headed, and doing the concrete work now to anticipate them and intervene. It's very difficult to do. But I think, Australian regulators need to be applauded for the work that has been done here, in contrast to China, in contrast to the US. And I think what will end up happening is that things are going to get pioneered elsewhere. Things like the Novartis leukemia treatment that costs $400,000 on the open market. And now, parents who have children with leukemia, parents who have babies with spinal muscular atrophy and really desire that $2.1 million treatment, they're lobbying the government right now to change the, regulations of the national healthcare system. And, the question becomes, is the healthcare system going to collapse under the weight of these new genetic therapies? And I think that needs to be another part of where, collective action can be happening now to anticipate, there's no reason why those therapies need to be attached to those price tags. The main rationale is that, the companies have been doing research for a long time to develop the therapies, but a lot of the research has been publicly funded as well. So some of the same conversations that we're having about intellectual property in vaccines need to be expanded to those these big high-priced drugs, because they are going to destroy the healthcare system in Australia, in Europe, even in these markets where it's not sort of insurance agents mediating who lives and dies, but it's more collective decisions about, what kinds of medicine can we afford as we move forward as a society.
Okay, thank you very much.
I thought I saw a hand up, but I think it was my cursor. And I've actually got a lunch date, so I need to bounce off. But
so much of your time. Thanks, Eben. I had a question, but I'm going to just email that to you.
Well, I can do it really quick. If we can, my lunch date isn't for another 10 minutes. And
okay. Well, I was just curious about whether disability rights activists have weighed in on this issue, because you give us a fantastic overview of how a whole bunch of, critical diseases like HIV/AIDS, leukemia, etc., can be addressed, and the ethical debate surrounding all of this. But I was wondering whether disability rights activists have weighed in.
Yes, for sure.
And if so, in what ways?
So one of my chapters is a profile of Gregor Wohlbrain. He's a biochemist and bioethicist from Canada, originally from Germany, and his body looks really different than ours. He is the first to tell you that he's very happy with his body. He doesn't want to have a body like ours. His mom took thalidomide when she was pregnant, so his arms and his feet and hands are different. And he's accomplished. He's an accomplished bench scientist and also a very articulate theorist. And part of his concerns are, he doesn't have a genetic condition, but his concerns are about societal norms about ableism and being able-bodied, and having that be kind of the marker of who gets societal resources, who gets access to employment. He asked questions like, will people like me be edited out of existence? And part of the book is also an exploration of existing genetic testing technologies that are in the clinic. So for decades now, we've had amniocentesis, but in the last 5 years or so, we've got a more ubiquitous testing protocol that many doctors are encouraging all pregnant mothers to get. So there's a non-invasive prenatal blood test that lets you see if your child has Down syndrome or if it has Klinefelter syndrome, a condition that impacts about 1 in 500 boys. So Klinefelter syndrome is basically an androgynous condition. And, you know, do we want to medicalize these babies and say that, you know, non-conforming gender identities are something we're going to eliminate from the future population? That's happening right now. So these decisions about, you know, aborting a fetus or continuing with a pregnancy are being delegated to clinicians and parents. And clinicians often don't have the background in genetic counseling to even, you know, they might have read about Klinefelter syndrome when they were in med school, but they might not have, you know, known that they were examining somebody with this condition. So there's a long history of disagreement between the medical community and the disabled community about what constitutes illness and a medical condition that needs fixing. So yeah, there's a chapter again in the book on Gregor, and he also features in a fabulous movie called Fixed that explores not only these questions as they relate to genetics, but also other kinds of technological enhancements and prosthetics. You're muted.
Thank you so much for that, Eben. I can't wait to get into your book. Your presentation has truly sort of, you know, triggered the juices for your book in me. I can't wait to get into it. Where can people? I mean, I'm sure it's available in the library, but do you want to just tell people where to find your book?
Yeah, it's available everywhere books are sold these days. I mean, it's out here in Australia from Black Ink, and I'm surprised all the corner bookstores I'm finding it in. And in the UK, it's fresh out in paperback. It's also on Audible and Kindle, so all the usual places.
Great, that's perfect. Congratulations again, Eben. Thank you so much for your time today. I really
Thanks, Yamini.
appreciate
Thanks
it so
everybody
much.
for the questions.